Yeah, it's a real pleasure to introduce Stefan Knapp. So as I was just explaining to him, the goal of Working Group 3, which is co-led by Carles Gaudiano and myself, is to try and promote directed proteolysis approaches by building bridges between biologists and chemists on a kind of EU-wide basis. And so it's fantastic that we have Stefan out to give us a talk, because he sort of embodies what we're trying to achieve. He is himself a bridge between that kind of chemistry and biology communities in this area. So he did his undergraduate studies in chemistry, followed by a PhD in structural protein crystallography at the Karolinska Institute. And since then, he's made his career very much at the interface between these two disciplines, both in industry and in academia, including as Professor of Structural Biology at the Nuffield Department of Clinical Medicine in Oxford, and as one of the directors at the Target Discovery Institute there, and then more recently at Goethe University in Frankfurt, where he's currently Professor of Pharmaceutical Chemistry and CSO of the Structural Genomics Consortium node there. Very well known for his work in structure-guided drug development, particularly, I think, protein kinases, epigenetic modifiers. But in recent years, he has turned some of his attention to the Ubiquitin system, and we're really looking forward to what he's got to tell us about that. His talk is about development and characterization of chemical probes targeting the Ubiquitin system. So we would encourage you all to ask lots of questions. Please post them in the chat, or keep them to the end of the seminar, at which point you can raise your hand and unmute yourself to ask directly. And we would also like to have a kind of informal discussion following on from the seminar. So I think we suggest that once we've finished the Q&A session with Stefan, we'll take a short break and go and grab a coffee or something. And then those who would like to, please do come back and join us for a more informal chat around topics that have been raised during the seminar, or indeed any other topics that you might like to raise. Okay, so I will now hand over to Stefan.
Okay, thanks a lot for the introduction. I just try to share my screen. Do you see my slides?
We do.
Okay. So I thought to start off, I give you a little bit of background, because our lab is, of course, an academic group in Frankfurt, embedded in the university. So my group is in pharmacy, but the last 20 years now, scary, that's already 20 years, we have been setting up a public-private partnership called the Structural Genomics Consortium. So at the moment, we are about 200 scientists working at different universities, Toronto, Frankfurt, Chapel Hill, McGill, Karolinska Institute, and the UCL in London. We collaborate intensively with pharmaceutical industry. You see the current funders at the bottom of the slide. And our main mandate is to solve basic science questions, problems that would lead to more efficient drug discovery. So we started off as a purely structure-focused initiative. That's why we call it the Structural Genomics Consortium. In 2004, our main mandate was to make structures relevant to human health. So far, we solved 4,000 new protein structures, also a lot with ligands. So that gave us a very good repository of structural experimental data. Then later on, we started to use these structures ourselves and develop bioactive chemical tools. So we call this chemical probes. And this is a program that merged now into our current funding scheme by the EU called UBOpen, which has the mandate to build a reference library for the trackable proteome. And in addition to that, we also use patient-derived cell assay systems to test these inhibitors and chemical modulators, and recently preparing for our next phase in 2025 is that we would like to move some of the design of small molecules to a machine learning and AI-driven initiative. So we could use this because in these 20 years, we realized that if we would like to cover the trackable proteome with high-quality small molecules, and we have this Target 2035 initiative. Also, this is a webinar where we very often invite people in the field. So please check the webpage and look if something interesting for you in there. If you continue with the current speed, that definitely will not finish in my lifetime to cover the proteome. So I think we need to find new technology. And I mean, for the protein structure, I think with the development of AlphaFold 2, the AI community used this large repository of experimental structures to make predictions that are now very accurate. Still not accurate enough to make reliable small molecule predictions. And we believe that developing more efficient small molecules will in the future have, of course, a large AI and machine learning driven component. So this will be some of the things we want to do in the future. And also our development here with the private funders, you see that the number of companies increased with time, and it really increased when we decided to be an open science lab. So that means all our data and all our reagents will be made available. And we don't file patents our own. So we do pre-competitive research, and that makes it easy to collaborate with 7 or 8 companies because they know that what we do would not be exclusive to that collaboration. So we can discuss about the system. I think it's interesting. Also leads to new drugs. I mean, I usually use the epigenetic area that we started in 2010, essentially, where we had no inhibitors for this bromodomain family. That was the main focus when I was still in Oxford. The last at least 5 to 6 years, we covered this entire family with small molecules that were exclusively selective and very potent. Particularly the beta inhibitors led so far to 44 clinical studies. You see our original molecule, JQ1, developed with Dana-Farber, led to clinical compounds shown below. One of them will be soon approved. So the phase 3 studies for a molecule looking very similar to this constellation molecule down here was found beneficial for myofibrosis in combination with JAK inhibitors. Here's another example. So this is our probe. This is the clinical compound that is currently in 2 clinical trials in phase 2. So you see the bromodomain binding moiety, this dimethyl isoxazole with the linking benzimidazole ring system came from the probe. The decoration, of course, changed. So we believe it's enabling. It's relatively straightforward for us to collaborate with the academic as well as with the private research, and we try to promote chemistry in or small molecule development and chemical tools. We see, and this is not an article by us, but I found it quite entertaining. If you look at the field, unfortunately, a lot of low quality molecules that are all bioactive because many of them are natural products, but they're not meaningful as chemical tools. Just to showcase this curcumin molecule, medicinal chemist will spot, of course, this is a Michael acceptor. It's highly reactive. I checked last year, I updated this, there are 22,000 publications using this molecule for very diverse areas. So it's not useful as a chemical tool. There are also 120 clinical trials. We eat this. This is the main component of the yellow color in curry. So it's not dangerous, but nevertheless, I think it is not a good idea using it in the clinic because it stays in your gut. And so the number of publications still increases in this area, but we believe that with continuous teaching what is a good chemical tool, we may convince the community to use high quality molecules where they can get mechanistic insight. And therefore we defined probe criteria for the small molecules together with the pharmaceutical industry that would allow us to develop small molecules that give mechanistic insight. So when you see a phenotypic response in your assay of choice, that would mean that you can also link it to the target because we comprehensively evaluate them. They are also potent small molecules. And this is another initiative, the chemicalprobes.org portal. It's a crowdsourcing initiative where the chemical probes are discussed openly, so people can leave comments when they find unexpected behaviors of small molecules. They're reviewed by the community. I think this is a good source if you look for your favorite protein molecule for a small modulator. And unfortunately, our effort did not always translate into this great teaching experience. Here is just the compliance of the community using our chemical probes in publications. So there are many thousand publications. However, one thing that we very often do is to provide a maximum concentration where a chemical probe is meaningful to use. However, you see that for some there is only a very, very poor compliance. It's one of the problems that biologists may tend to put a high concentration of a compound on cells to see a phenotypic response, but then you're leaving the window where we guarantee that the molecule is selective, at least within the family. Okay, all small molecules are also evaluated for their properties in cellular assays. So we would like to know if they are toxic, when they are toxic, why are the cells potentially dying. One of the main problems is precipitation, which is incredibly toxic for cells. So we look at small molecules that cause precipitation in cell culture and provide concentration when this happens. We look for fluorescent compounds for membrane integrity, nuclear integrity, fragmentation, binding to tubulins, mitochondrial and ROS signaling effects, and so on. That means in addition to the biochemical and characterization of the small molecules, we also characterize them in cells when they go into our probe and what we call chemogenomic libraries. Okay, coming to the degrader field. So when I saw the slides, I was not sure how much you work in that area. So now it turns out that I probably can be quite quick here. Everybody probably knows the story of thalidomide, particularly in Germany, where I think it was the largest pharmaceutical scandal we had in the 50s. This molecule was developed, marketed under the name Contergan, was for a long time in use, 3-4 years. Several thousand children have been born with defects on their limbs, ears, and so on. And it took actually 50 years to find out why. The reason is that this molecule binds to a E3 ligase called cereblon. Here's the science paper 2010 that reported this and acts then as a molecular glue, degrading transcription factors that are very important for limb formation. So we can, with small molecules now, make two types of bioactive compounds. So either glues, where we can recruit in part very untrackable proteins, such as transcription factors, and selectively degrade them. And for this, we have now already several examples based on thalidomide and their derivatives in the clinic, where we design in principle quite randomly decorations that go outside of the E3 binding surface and try to recruit different proteins like this Icarus family, GSPT1 is in cancer clinical trials, and so on. and make molecular clues. The design of this is still a trial and error. It's quite difficult to make them. More rational it is to rationally link two inhibitors, one binding to E3 and the other binds to a certain target protein. And we call this proteolysis targeting chimeras or PROTACs. And at the moment, there are several clinical studies ongoing that develop these chimeric molecules. So we find them quite exciting. Here's the mode of action. So we link a E3 ligand with a protein of interest ligand. This allows us to recruit the protein of interest to the ubiquitin system. Here's the complex of the E3 and the E2. The yellow circles is the ubiquitin. It can be then transferred to the target protein, causing degradation, and the complete protein is degraded by the proteasome. And after that, the PROTAC can enter a new cycle, degrading another protein. And that means that we have here a molecule that acts catalytically on the degradation of a protein that is causing a certain disease. And so there is, I think, a really great opportunity to develop a new type of pharmacology. So it has now been named event-driven pharmacology, because this is a catalytic agent that we are developing. We can target all protein functions, not only enzymatic inhibition, that means also scaffolding functions. In part, at least, we can address undruggable targets, that as long as we can bind to the surface somewhere a molecule with sufficient affinity. And I talk a little bit about this issue of tissue specificity. If we would now select the E3 ligase, at the moment we are still lacking ligands for that, but I talk about how we develop them. We would be able to degrade tissue selectively a certain protein of interest, maybe just in a diseased tissue, particularly in cancer. There are many cases where E3 ligases are highly expressed that we could target and therefore make cancer cell-specific drugs. However, the molecules get larger. If you link them together, you usually end up with molecules that are definitely larger than conventional drugs. There is a context dependency, also depending on the E3 expression level, potentially also a mechanism of resistance. If the E3 is not required for a certain tissue type, it may actually be silenced and causing resistance of the degradation. Then the pipeline is less established, and I talk about this issue, how we develop assay systems to rationally develop PROTACs. And then that also means, if you look from a tool perspective, what is really a good PROTAC? So that's a little bit hard. For enzymes, you have a EC50 and an IC50, and the selectivity. So with PROTACs, we make at one time point a mass spec. So the PROTAC action is concentration dependent, it is cell type dependent, and it is definitely also dependent on kinetic factors such as turnover from proteins that are targeted. So I think this is at the moment a discussion in the community, what constitutes a good tool and also what constitutes a good drug in that context. So in contrast to a small molecule to develop for an enzyme, developing a PROTAC needs a lot of assays if you really want to find out where the problem is in this cascade. And here is different assay systems that we develop. So first of all, your molecules are pretty large, so they need to go to membranes. We measure this using a Brett assay, and I explain this in a minute. So we make assays that look at the binary complex formation. That means the target engagement of our PROTAC with the protein of interest and with the E3. And we can lyse the cells at least partially. So we take digitonin that makes holes into the cell membrane. And that allows us to evaluate how much the cell membrane is a barrier for these larger than usual small molecules, and if we efficiently reach our target. Then specific for the ProteoCURE is this so-called ternary complex, and the ternary complex is between the protein of interest, here is POI, the ProteoCURE, and the E3 ligase. So this triggers degradation. So the field believes at the moment that the stability of the ternary complex is one of the main bottlenecks creating efficient ProteoCURes. So you may get away with weak binding to your target, but you need efficient ternary complex formation that is maybe also long lasting in order to ubiquitinylate the protein of interest for degradation. And then once it's ubiquitinated, actually, and these things, they just happen. We made sensors for these systems that allow us to see how much ubiquitinylation we get and if the proteasome is recruited. But this is the typical degradation machinery of the cell. Once you have ubiquitinated your protein appropriately with the right ubiquitin lengths, of course, this again is a problem of the ternary complex, you get definitely degradation of your target. So, and everybody in the field knows this so-called hook effect. That means at a certain concentration, here's one example of our developed ProteoCURes based on a inhibitor called alisertib that binds to aurora kinase. You see that at 10 nanomole you have significant degradation of the target. At 100 nanomole you barely see it. At 1 micromole the band gets again a little bit more intense, and at 10 micromole it's back. And this is that because at high concentration the ProteoCURe concentration also increases in the cytoplasm, and that means you preferentially get binary complexes and not ternary complexes because of competition. And your proteins then are no longer degraded. That means just looking at the equilibrium thermodynamics of this, that cooperative synergistic ternary complex formation would actually prevent the hook effect, because you could have weak interaction with the target of interest and with the E3. If you would form synergistically a ternary complex, you would never compete for the binary complexes at high protein concentration. So we looked at different systems to develop assays for this. Here's an example where we had 3 different series of ProteoCURes, 2 developed by us, and one was from the Lab, and he made the SAR series available to us. You see one molecule for this protein of interest called WDR5 has been developed as a SGC chemical tool. You see that here. This is a published compound that we used from the literature, also a strong binder to WDR5. And this is a third series of ProteoCURes based on different decorations on this original scaffold that is more potent, called MS67 from the Lab. So all these molecules have been developed with different exit vectors to link it to the E3. So we thought it's a good system to study the active and the inactive compounds of this series to see where the problem really is and why we do see degradation for some of these ProteoCURes and not for others. So here is the binary complex assay. So we fuse our target of interest and also the E3 with a nanoluciferase, which is a highly fluorescent luciferase, in frame with our protein of interest, in that case WDR5. We make a tricer molecule that looks similar than our chemical probe. Here's the tracer that we synthesized. We have here this BodyPie type of fluorescent tracer linked with a PEG linker to our protein of interest ligand. Now, if you are close to the luciferase, that disappears when you compete away your tracer with your ProteoCURE, so you can measure dose dependently the stabilization of the ProteoCURE WDR5 complex that is concentration dependent. So this is in intact cells, this is in lysed cells. These are the parent molecules. And if you compare it now with the ProteoCURE, you see that you get a significant shift to the right for the entire series, whereas the lysed or permanentized cells behave similar than the parent ligand. So we usually see a tenfold or even more difference in intact cells because of the size of these molecules. And some of these molecules did interact really weakly. For instance, this compound, JW48, shows almost no interaction that we could measure even in lysed cells. We could measure this by SPR. It's in the high micromolar range, but nevertheless, we thought we carried this ProteoCURE along in our analysis, and I show you later some quite interesting data why this molecule still work. So then the ternary complex that is really critical, we assemble with a dual tag system on the target of interest or on the VHL. We have two fluorescent groups. One uses the nanolook, the other one uses the halotag. And the halotag can be easily functionalized by a halogen alkane. So it's a halogenase, a bacterial halogenase that is mutated. So when you have a halogen alkane, it forms a covalent link with this halogen tag. That means you can take a dye molecule or anything else you want to functionalize here on this tag. In cells, you get a covalent linkage to the halogen tag. And in this case, we use the tracer molecule, very similar to the ones that we use in our binary Brett assay to create a Brett pair between a dye conjugate of the halogen tag and the nanolook system. So we can sense the formation of the ternary complex. We can make washout experiments and look at the stability of the ternary complex in live cells, looking at the dissociation when we wash out the ProteoCURE. So first of all, we need to play around a lot with the tag positioning. So here the N and we have the tags at the N terminus that did not give us a good signal. For instance, VHL is the E3 at the N terminal tagged with Brett and halotag on the WDR5 N terminus. There's almost no signal available. Here, taking this around, the best signal we get with the N terminal VHL halotag and a C terminal W's DR5 halotag. You see that we get a very nice Brett signal from this pair. So there's quite some cloning involved to optimize the assays. And we could rationalize that as well, looking at the ternary complex crystal structure, where indeed the C terminus and the N terminus in that arrangement are in relatively close proximity that allows a Brett signal that is induced here by the ProteoCURE that brings the E3 in contact with the WDR5. Okay, so looking at the three different series, we get very different signals in Brett, and that means that the orientation and the distance of our sensors is different for all these. So we have here a high Brett signal for this MS67 ProteoCURE, which of course would correspond to a certain conformation in solution and the highest proximity. We have a medium Brett signal, and here for this the distance is too far to measure functional Brett. And then we have a medium low Brett with the highest complex distance in the three conformations, and we made crystal structures of most of them and could validate that. It's a little bit critical doing this by crystallography because the orientation of these molecules here might be influenced by crystal contacts. But if you take them as face value and hope that will be indeed the conformation you see in solution, looking at where we put our labels, it is clear that the conformation 1 has the closest proximity of the nanolook and the halo tag that we've used here to these proteins. So we use another system that allows us to monitor the protein levels in cells, and this is a reconstitution system which is called hybrid. In the hybrid system, we just use a small peptide that is called the hybrid infusion with WDR5. So we use CRISPR to CRISPR this into the locus, in this case the C-terminus of WDR5. And the rest of the nanoluciferase, the large bit, we can express in the cells or add it to a buffer. So with this we can reconstitute a functional luciferase molecule, and together with a tracer we can also use this system to measure degradation, or we can just use the tag to measure the kinetics of degradation. And this is also the system that we used to look if our protein of interest are recruited to the proteasome and that are ubiquitinated. You get a degradation kinetics, so we can follow in live cells the level of the protein by simply looking at the fluorescent signal that we get, or the luminescent signal that we get from our luciferase. You see here, this is the negative control. There's no effect on protein level. Whereas here we see that after about 20 hours, you reach a steady state, and we can look at a very long time point that the steady state is stable. So we believe that is probably the equilibrium between resynthesis of our target and the degradation that we get for this ProteoCURE. And that means this system allows us to determine two important parameters. One is the so-called Dmax, so that's the maximum level of degradation that we get. It's hard to do that by Western because it's so dependent on expression levels. Some of the targets may be barely detectable, so it's very hard to judge if there's really nothing left in your Western plots. With this system, I think we can be quite confident how much is left, so it's not completely degraded. And we also get EC50, which is the equivalent to the EC50 for an enzyme inhibitor, and this is the concentration where we see the half concentration level. So we can do this now for all our projects, determine the DC50 as well as the Dmax for these and characterize these small molecules. And we usually also use either a methylation or a proteasome inhibitor as a positive control to show that this degradation is really on target. So we are dependent on the proteasome in that case for the degradation of this molecule and not maybe on transcription or any other process. So the exciting information here is that, of course, you have some products that just work on all assays. They form stable dimer complexes. They form stable ternary complexes. They have good DC50. They have very good Dmax, more than 80% in that case. This is another series we developed a little bit weaker in terms of the DC50 and the Dmax, but we see they form binary and ternary complexes in cells. But we also found molecules like this where we don't detect any binary complex formation in cells, only very weakly in lysed cells. But we saw that there is strong ternary complex formation leading to DC50 and a very nice Dmax. So that would, I think, be an ideal molecule to develop further. Of course, we would need to improve the Dmax, but here we really have a a weak target affinity, so there is no interference really with binding of this inhibitor to our WDR5, because this interaction is very, very weak. We have a high synergy in the ternary complex formation and therefore get a very efficient degradation of the small molecule. So this is a whole lot of assays we need to set up to really characterize small molecules that are degraders, and also to improve them further. We think it's worthwhile doing this exercise because we find classical Western blotting that we also do for the main lead compounds in the end are poorly adapted to high throughput screening. So if we need to make hundreds of molecules tested at different time points, maybe at different concentrations, of course, that is a whole lot of Westerns that have a high variability and very prone to problems such as transfer and protein levels itself. So coming to the E3 family, so these are multi-subunit E3 ligases. You know, it's a huge family, a total around 600 in humans. On these multi-subunit E3 ligases, there are a number of ligands that have been developed. In our hands, this product here is highly unstable and reactive. We don't use this. These are covalent ligands to E3. This idea is great because if you covalently link your molecule to the to a E3 ligase, you would reprogram this E3 ligase to degrade a certain target molecule. However, this electrophile used, at least in our hands, is very promiscuous and too reactive. It reacts with a large number of proteins in the cellular context, so we don't use them. So that just leaves us with very few ligands that we can use in chemical biology for this huge family of E3 ligases at the moment. So I think there's enormous potential for further development. And also this family that contains, for instance, the TRIM proteins and the Kelch proteins is preferentially expressed in certain diseases and certain tissues. For instance, this here shows the expression here of in different types of cancer cells, and we see that particular this family here would be fantastic for expressing, for instance, here TRIM15 only expressed in the intestine and duodenum, so very selectively we could make degraders for this type of tissues. Here's some more cancer cell lines. This is the red ones are up to 20-fold overexpressed in these cancers. And if we would be able to make small molecule ligands for them, we would be able to selectively degrade certain cross-promoting factors in these cancer cells without affecting the other tissue types. Okay, how we do this? So it's just one experiment I show you here. So we have many screening initiatives. One is used DNA encoded libraries, and another initiative you do quite extensively is ASMS, so mass spectrometry based screening technology. Here we use two types of libraries. They're all commercially accessible through companies. However, the structures of these molecules are not known, but we convinced the companies to put the fingerprints of the structures at least into the public domain, so we can use machine learning to make predictions of public libraries, and all these molecules and the fingerprints are available in a database that we just setting up. It's called AirCheck. So you can get access, you can screen your own libraries with the machine learning programs, make your predictions, and then just purchase the ligands. It also overcomes one of the limitations of DNA encoded libraries, which is the chemistry in the presence of DNA molecules, which is very tricky because DNA is very reactive. And if you just use the fingerprints, you can also get molecules that are less dependent on synthetic constraints you have if you design DNA-encoded library screens. So here's an experiment. So it's a number of WDR domains that are also, we have about 100 E3 ligases with this domain. For instance, DCAF1 is one of them. And we screened now many of them. Here you see the hits that we got in our machine learning fingerprint search technology. So there were several new ligands identified. This is just being published. I think is 30 screens of WD domains against 2 × 10⁸ molecules, together with the data that are in this AirCheck database. So one example where we identified for E3 ligase a ligand that was DCAF1. The initial hit was a ligand already having considerable activity, about 490 nanomole. Here's the crystal structure. You see the ligand has now been published in Journal of MedChem by the SGC in Toronto. And just want to finish up with a second story where we use ASMS screening, identified a ligand and developed into a chemical probe, because this paper will be published hopefully next month in Nature Chemical Biology. And that was a collaboration with Pfizer. So these are the initial hits that we found by screening, and this is the final molecule. So it targets N-terminal decoron E3 ligase called GIT4. From yeast, we know that this ligase should actually regulate metabolism. There are many gluconeogenic enzymes in yeast that are regulated by this decoron, which contains the N-terminal proline. That's still part of the structure of the probe. So we developed the molecule based on the consensus sequence and the screening data that we got. That is now called PFI7, has a KD of 80 nanomole. Here's the crystal structure and the binding mode of this molecule. And so we are targeting the GIT domain, which is a beta propeller domain. You see the structure here as a ribbon diagram that recognizes the decoron sequence. And here's the biochemical and SPR data. And we usually make negative controls. So when we introduce a metal group here at this linking CH2, we get an inactive control compound because of steric exclusion. And this can be used as a control also for the development of ProteoCURE. So first of all, we had to set up assays. So I already mentioned we do this usually by PRET technology. So here's the chemical tool with a PEC linker, and we have the dye attached. This is the PRET titration. So we can now measure the interaction in cells. You see that we have about 300 nanomole, depending if we N or C terminal link the GIT4 with the nano look. So we use this as a sensor in cells now, and we did a BioID study, which is a system that allows us to detect interaction partners that are between 100 and 200 nanometer in the neighborhood of our protein of interest. We do this by biotinylation transfer, recruiting a biotin transferring enzyme to a small tag that we also use in CRISPR technology to put into the GIT4 locus. So with streptavidin, we can look at the peptides and see what complexes we recruit. So to our surprise, in human cells, the GIT4 is not involved in metabolism. It seems to be a protein or E3 ligase that is involved in the regulating proteins in the nucleus, in particular chromatin binding domains at ribonucleoproteins, spliceosome complexes, DNA replication, and so on. So I think it probably shifted function between yeast and human. We also looked at interaction partners via the Git domain, and saw that the consensus sequence in human is less pronounced than in yeast. Here's the consensus that we got. Still contains the proline, also conserved in yeast. And we particularly were interested in a number of helicases that strongly interacts with Git. See here the DDX55 and DDX21. These are helicases. KIF is a small motor protein transporter of the kinesin family. And here you see the top hits of our interaction study that we did on this. And at the moment, we are using this to make ProteoCURE, of course. So we made now several adducts with, for instance, our JQ1 molecule, which is very well established as a highly degradable protein called BRD4. So we made about 10 of them. We are testing them now in our sensor cell lines, and as well as other molecules like HDAC inhibitors, so we can test the whole family by fusing here SAHA, which is a clinical compound that interacts with all HDACs, to see if we can get functional degradation of any of these using this E3 ligase in combination with a protein of interest ligand. Okay, this is what I wanted to share with you. So our group, of course, there's a group working on the chemistry, which is led by Vaclav. And there's another chemistry team that more works on the protein of interest ligands. I haven't talked about protein kinases, which is one of the main areas still in the lab, developing small molecules. So we combine this also to make now degraders. There's structural biologists and biochemists that allow us to make complexes. Andreas Krämer did all the structural work on the WDR5, and Martin Schwalm did all the assay work on this and the sensors. There's a lot of collaborators, probably too many to mention. And particularly want to highlight two, because I talked a lot about WDR5. This was with Jianjin's lab and Mount Sinai, who donated to us one of his lead series, so we can also include them in our study. And Promega, who is co-developing with us many of the tracer molecules, which are then made available to the community via this vendor, so we don't need to resynthesize them and send it out ourselves. And of course, funding in the German side is now funded by the UB Open Consortium mainly, as well as other academic grants. I listed them here. These are mainly project grants, but also ProxyTrucks, which is a large government-funded initiative developing new types of degraders here in Frankfurt. So I hope that gives you a good overview, and also about this field and our work, and I'm very happy to answer any questions you may have on this topic.
Thank you very much, Stefan. It was a really, really interesting, fascinating talk, and given us a lot to think about. So